The Prix Galien is a welcome initiative to stimulate creative research and promote excellence. Barack Obama. The Galien Awards ceremony is considered the equivalent of the Oscars night for the innovators in the labs and awards every year the best pharmaceutical product, the best biotechnology product, the best medical technology, and the best digital health product.
This is the right event, on the right issue, at the right time.
I thank His Excellency, the President, for bringing Prix Galien to Africa, and I look forward to the day when we will celebrate an African winner of the Prix Galien.
I'm particularly grateful to receive this award.
The awards are among the highest honors in science and commerce because they lead to improvement in the human condition.
The Prix Galien Awards recognize the world's brightest minds and most innovative companies. They are a true celebration of the hard work required to produce life-changing interventions.
That is what makes us optimistic about the future.
Congratulations to all of you.
Make a difference. Join the Galien Foundation. The Galien Week of Innovation has been made possible thanks to our precious friends and partners who share the Galien Foundation's values. First off, the two exclusive platinum partners are We would now like to acknowledge our two gold partners. Furthermore, we proudly introduce our corporate leaders partners. The associate partners are the grantors are To conclude, we are extremely fortunate to work with outstanding media partners who have contributed to the Galien Foundation's renown.
Good morning, everyone. I'm Kyle Bryan, the Chief Medical Officer of IQVIA Biotech, and I'm pleased to welcome you to this, the third day of the Galien Foundation Week of Innovation webinars, held in conjunction with the annual Galien Forum of Speakers representing top scientific experts and leaders in global health. This year's Galien USA Award set a record with 84 nominees from 65 companies covering 15 diverse therapeutic areas, running the gamut from cancer to cardiology, neuroscience to infectious disease, and highlighting some very rare genetic disorders. We've been looking closely at these products in all the critical areas throughout the week, and we're excited to continue today. Because of the number of nominees and the range of conditions represented by this excellent science, the Galien USA Awards Committee decided to host this series of 14 webinars leading up to Thursday's forum and awards ceremony, where the winners in each of the 4 best of categories will be announced. The objective of this series has been to give greater visibility to the nominees in our categories of best pharmaceutical product, best biotech product, best medtech product, and best digital solution product, and to give the attendees an opportunity to hear insights describing the discovery and drug development process. In the best pharmaceutical agent category, which is our focus today, we're holding 6 webinars to discuss the 33 products representing this important field. This hour is devoted to 4 of the nominees in solid tumor malignancies. Our topic today relates to this central question. What's the state of progress in building new therapeutic and diagnostic platforms to fight major cancers and to address their prevalence in vulnerable populations, including the aged, minorities, and pediatric patients? Please note this webinar is for you, our audience, and it includes time for some of your questions. So I would encourage you to forward your queries to us using the link provided on your screen. Now, before moving into our discussion, I'd like to take a moment to get the audience input on a quick poll. What is the most promising new medical advance likely within the next 5 to 10 years? Would that be effective and accessible vaccines for major global killers like HIV, malaria, and TB? Solving the blood-brain barriers leads to advances against a variety of neurologic disorders, including clinically effective treatments for Alzheimer's. The introduction of comprehensive diagnostics and therapeutic vaccines as a preventive tool against a wide variety of cancers. Further refinement of safe, clinically effective CAR-T and other cell or gene editing therapies across tissue types and disease conditions. Or widespread use of patient-friendly digital and biosensor platform technologies that control or prevent the progression of major chronic diseases. So, let's move on to our discussion. I'd like to start by introducing the 4 nominees representatives who are joining me for this webcast. They are Dr. Rodrigo Ruiz Soto, Senior Vice President of Clinical Development and Translational Medicine for Deciphera. Deciphera has been nominated for Kinlock, a novel switch-control tyrosine kinase inhibitor designed to broadly inhibit KIT and PDGFRA mutated kinases through a dual mechanism of action and indicated for the treatment of gastrointestinal stromal tumors. We also have Dr. Jason Brubaker, Vice President of Medicinal Chemistry at Blueprint Medicines. Blueprint has been nominated for Gavreto, a kinase inhibitor of wild-type RET and oncogenic RET mutations and fusions for the treatment of RET fusion-positive non-small cell lung cancer and thyroid cancer, and for RET mutant thyroid cancer. We have Dr. Ian Webb, the head of U.S. Medical Affairs for Bayer Oncology. Bayer has been nominated for Nubeqa, a first-in-class oral TRK inhibitor for patients with non-metastatic castrate-resistant prostate cancer. And we also have Dr. Barinder Kang, Vice President and Head of U.S. Medical Oncology and Radioligand Therapy for Novartis. Novartis has been nominated for the development of Lutathera, a peptide receptor radionuclide therapy targeting the somatostatin receptor for the treatment of cancerous neuroendocrine tumors affecting the pancreas or gastrointestinal tract. Before we go to our panelists and our main topic, I'd like for you to note that there's an accessible link to a brief statement submitted by the companies on each of these nominated products. And this link is available for viewing during this webcast. So moving on to our main topic today and our panelists. As we all know, cancer is a devastating disease. It directly impacts the lives of millions, with deaths in the hundreds of thousands every year. One of the primary lessons that we've learned in the fight against cancer has been the realization that this is a heterogeneous disease, and thus our standard treatments like chemotherapy and radiation work only for a relatively small subset of the patient population. The biology of these malignancies can have quite different underlying genetic and environmental causes. And similarly, these tumors can express very different proteins in one patient versus another. This intrinsic variability in cancer lends itself to this ever-expanding field of precision and personalized medicine, characterized by the pharma products, such as the ones we're discussing today. It's increasingly clear that precision cancer treatments like these can result in immense patient benefits. It's particularly true when we talk about vulnerable populations like the very old or very young, as well as the patients who continue to be underrepresented in both our clinical studies and in available therapy, and thus aren't getting the full benefit of our advances. There's a recognition that this kind of product development using precision cuts rather than broad swaths, both to our industry and to regulatory agencies. But we've still got a long way to go. There's going to need to be broader changes to the healthcare and insurance systems if we're going to fully realize the potential of personalized medicine in cancer care. So I'd like to open our discussion by asking each of our panelists to share his thoughts about his drug product and the path to development. Rodrigo, I'd like to start with you.
Yeah, good morning and thank you for the invitation. So gastrointestinal stromal tumors, or GIST, are rare sarcomas and the most common sarcomas of the gastrointestinal tract, with an annual incidence of between 4,000 and 6,000 new cases in the US. About 80% of GIST have a primary driver mutations in KIT and 5 to 10% in PDGFR-alpha. Advanced GIST is marked by the development of secondary drug-resistant mutations, which play a key role in disease progression and drive resistance to established therapy. The standard of care for patients with GIST are tyrosine kinase inhibitors. Imatinib is used in the first line, sunitinib is used in the second line, and regorafenib in the third line. There was a significant unmet medical need for patients in the fourth line or later with no approved therapies and a need for broad spectrum inhibition of KIT and PDGFR-alpha primary and secondary kinase mutations. QINLOCK is a novel switch-control tyrosine kinase inhibitor specifically designed to treat GIST patients using the Cyphera switch-control kinase inhibition platform, and it was developed to broadly inhibit KIT and PDGFR-alpha mutated kinases with a unique dual mechanism of action. Invictus was an international multi-center randomized placebo-controlled phase 3 clinical trial in GIST patients who have received 3 or more prior anti-cancer therapies. QINLOCK demonstrated a powerful progression-free survival benefit. The median PFS using a blinded independent radiological review of CT scans was 6.3 months for QINLOCK versus 1 month for placebo, reducing the risk of progression or death by 85%. Notably, the median progression-free survival of 6 months exceeds the historical data for both sunitinib that is used in second line and regorafenib in third line, which is less than 6 months in both cases. Patients receive a clinically meaningful survival benefit when treated with QINLOCK versus placebo, a median overall survival of 15.1 months versus 6.6 months, respectively. QINLOCK was also generally well tolerated. Most of the adverse events that we saw were grade 1 and grade 2, and it also showed a clinical relevant quality of life difference when compared to placebo. In a long-term follow-up analysis, QINLOCK continued to provide clinical meaningful benefit with a well-tolerated safety profile. QINLOCK has emerged as a new standard of care for patients with 4-line or later GIST, where there was no significant unmet medical need with no approved therapies. QINLOCK was FDA approved in 2020 and is category 1 NCCN recommended for preferred therapy, and it is consistently accepted by clinicians treating patients with GIST. One lesson that we learned in our experience when we were developing the drug is that for rare diseases like GIST, we don't always have good preclinical models to help predict success in the clinic. At the time where the project started, there was a lack of preclinical models available and the lack of thorough sequencing data of tumors with drug-resistant GIST that make it more difficult for us to design the preclinical experiments to ensure that the compounds that we were evaluating would be broadly used for all GIST patients. This required doing much more research, working closely with collaborators, academia, and engineering a number of preclinical models that aided in the selection of the drug to move forward into the clinic. Thank you.
Rodrigo. I appreciate that nice overview. Jason, may I ask you to go next?
Sure. Can you hear me okay? Okay. Great. First of all, I'd like to thank the Galien Foundation for the opportunity to join the discussion today. And it's great to have our story included in the panel along with the other innovations here today. So I'd like to talk briefly about our platform and why we chose to pursue a therapy to treat red-altered tumors. Blueprint Medicines, we're a global precision therapy company. We started operations around 2011. And our founder's idea was to build a high-quality kinase inhibitor library without having a particular target in mind, and to screen them upfront against the entire human kinome. And for audience background, kinases are key signaling proteins involved in most cellular processes, and they've been proven drivers of disease in certain cancers and other rare diseases. So the idea was that when a new kinase disease driver is discovered, our kinase inhibitor library can help us quickly identify promising starting points for a drug discovery program. So this novel platform was put to the test when RET alterations were discovered as a disease driver in non-small cell lung cancer around 2012. And this was a rare subset of non-small cell lung cancer patients, about 1 to 2%, that were believed to be driven by RET alterations. Now, at the time, there were several multi-targeted kinase inhibitors that were either approved or in development for other indications that had some activity against RET as an off-target. But there was a clear need for a therapy specifically designed for RET that could fully inhibit the primary disease driver with minimal off-target effects. And so since this was an area of high unmet need, where RET-targeted precision therapy had high potential to be effective, we started a drug discovery program in 2013 targeting RET. And our library provided two clear benefits. We could immediately start with high quality starting molecules, and we could prioritize them not only on their on target, but because we do kinome-wide screening, broadly on their off-target activity as well. This enabled us to start with multiple diverse series of compounds and rapidly progress one of them to the discovery of Gavreto. Because of our platform and our people, we were able to bring this therapy forward rapidly to serve a patient population in need. And this was a breakthrough because that population was previously unable to realize the benefits of precision medicine, as Kyle mentioned. So I'd like to briefly talk about the impact on patients. So we've been able to treat patients with RET alterations broadly, and there's been consistent clinical activity regardless of the type of RET alteration, tumor type, or previous lines of therapy. We've had particularly encouraging clinical benefits in the frontline setting or when patients haven't been treated with a prior therapy. In addition, something we did that was a little bit different at the time was we anticipated and designed for overcoming resistance mechanisms. So these are genetic escape mechanisms that cause the drug to no longer work. And we believe that this approach that others are pursuing now as well has led to more durable efficacy, deeper responses, including complete responses in subsets of patients. And importantly, because the medicine is well tolerated, patients are able to stay on therapy. Clinicians have responded favorably to this and highlighted that it really was previously an area of unmet need. So we've had several patient interactions here at Blueprint several times. And if I still have time, Kyle, I'd like to share briefly three stories that impacted me fairly deeply. So around 2015, 2016, right around when we disclosed that we were working on this drug discovery program, we were contacted by a patient, an older male with a family who had recently been diagnosed with RET-driven non-small cell lung cancer. And at the time, we weren't yet at the IND stage, so we corresponded with him every now and then. And he was telling us things like, there really are patients like me out there. We appreciate everything you do. And sobering thoughts such as, I'll try and live long enough to when you guys start a trial. So unfortunately, we didn't get there in time for him. As we started our trial, we reached out and he had already passed away. So for me, that impacted me deeply because I know we all work with urgency and we try to do things quickly, but sometimes it's not quick enough and these patients are waiting for us. So the second story, more optimistic, was around 2018 in development. We were able to have a patient visit from someone on one of our trials. Her and her family came by. And for me, that was like the first time that we got to see the actual impact on a patient, beyond beyond the waterfall plots, what was the impact on this patient's life and her family's life. And as well, it was rewarding to see her walk through our labs, meet some of the scientists that were directly involved with this discovery. At the same time, sobering as well, she shared her fear with us that I understand many cancer patients' fear is what's going to happen for me if this therapy stops working. And so for me, that's, again, a call to arms to the industry to continue to think deeply about why these patients progress and what are the next lines of therapy we can start to prepare. And then finally, after approval in early 2021, we had a story of a patient in Alaska that was shared with us. This was a mother who had just given birth at 34 weeks. And at the time of giving birth, she was diagnosed with metastatic lung cancer that was determined to be driven by RET. So at the time, she was about to be put into hospice when her physician got her genotyped. The physician reached out to us at Blueprint, and through some heroic efforts by our commercial and patient advocacy teams here and a pharmacist at a specialty pharmacy, they were able to get Gavreto right before a FedEx drop-off and get this patient on therapy 18 hours after her physician contacted us, and she went on therapy instead of going into hospice. And I don't know how that story has continued, but I like to think that she's now at least able to have a chance to see some of her child's milestones rather than going into hospice, which would have been the previous outcome before Gavreto. So that's all I'd like to share for now. Thank you.
Thank you, Jason. That was very interesting, and I always find the relation of the patient stories to be particularly impactful. There's a misconception, I think, among the lay public that those of us who are involved in drug development, those of us who are in the industry, are focused on it as an industry. And I've long said to everyone that the minute we lose sight of the patient, all the work that we're doing ceases to matter very much. So thank you for sharing those patient stories and bringing that lesson back to us so strongly. Ian, could I ask you to go next?
Certainly. So thank you to the Prix Galien for the opportunity to join the webinar today. Really appreciate the discussion so far and look forward to the rest of the program. So first of all, just want to share that the Bayer Oncology's focus is on addressing unmet medical needs. And one of those unmet medical needs is in patients with advanced prostate cancer. Despite recent advances in the treatment of castration-resistant prostate cancer that is not yet metastatic, there remains a high unmet need for therapeutic options that not only delay the time to metastases, but importantly also have a proven safety and tolerability profile, which is especially important in the elderly, such as the patients who typically are undergoing treatment for advanced prostate cancer. Nubeqa is a second-generation androgen receptor inhibitor that has been FDA approved for the treatment of patients with non-metastatic castration-resistant prostate cancer that both delays metastases and has also been proven to extend survival. And prolonging metastasis-free survival is a key therapeutic goal in itself, as development of metastasis in these patients increases morbidity and mortality and increases healthcare costs. There's also a need for treatment options that have limited drug-drug interactions in the elderly, which then allows greater flexibility for physicians to treat other comorbidities that these patients typically have, such as hypertension, diabetes, or cardiovascular disease. Some recent data that shares the outcomes and the efficacy of this drug and its impact include the data on overall survival, which was added to the label as part of a SNDA in January 2021 by the FDA. And so the prescribing information now indicates that Nubeqa significantly reduces the risk of death by approximately 31%. Importantly, there are other quality of life benefits for this drug. In a post hoc analysis of the primary registration trial, Nubeqa plus androgen deprivation therapy prolonged the time to first prostate cancer-related invasive procedure, which was an exploratory endpoint, such as locally invasive procedures for treatment of cancer within the prostate itself. And we're going to be working with our partner Orion Corporation to extend the development of Nubeqa to patients with metastatic disease, and there are 2 ongoing studies in that area. One looking at Nubeqa in combination with androgen deprivation therapy on its own, that's the ARA-NOTE study, as well as the ARA-SENSE study, which combines Nubeqa with docetaxel and androgen deprivation therapy. And importantly, both docetaxel and androgen deprivation therapy are standards of care in this patient population, and hopefully will continue to have an impact improving outcomes in these patients with advanced prostate cancer, so that they can enjoy their elder years without significant complications from the disease or from its therapy. So thank you again for the opportunity.
I appreciate that. Barinder, you could tell us a bit about Lutathera.
Good morning. Thank you, Brian. I think first of all, I'd like to thank the organizers from the Galien side, as well as Dr. Brian for hosting this webinar and also giving this opportunity to discuss Lutathera. I learned a lot from other panel members also on their journey with the treatments they are discussing today. So first of all, talking about the neuroendocrine tumors. So even though neuroendocrine tumors are considered to be the rare tumors or orphan disease, their incidence has grown over more than 500% over the last 3 decades. This is based on the data, a SEER database of 2014. So it's approximately, we get a little shy of 7 new cases per 100,000 per year in the US. And there are only few treatment options for adult patients with inoperable advanced somatostatin receptor positive GAPNet disease once they progress on somatostatin analogs. So a little bit about the platform. So Lutathera is, or we also, from the technical name, from a lutetium-177 dotatate, it's a precision target therapy approved by FDA in January 2018 for the treatment of somatostatin receptor positive GAPNets, including foregut, midgut, and hindgut neuroendocrine tumors in adults. So it belongs to a special class of treatment called peptide receptor radionuclide therapy, or PRRT. This was the first FDA-approved peptide receptor radionuclide therapy, which provided new treatment options for patients whose disease progresses on somatostatin analogs. As a platform, this provides a totally new way of treating cancer, where you are combining both imaging and treatment isotope to the same targeting ligand, and making it a targeted radiation therapy or a theranostic approach. So the approval was on the basis of phase 3 NETTER-1 study in Lutathera, where Lutathera 200 millicurie in combination with standard of care octreotide LAR was compared with high dose octreotide. And the primary endpoint was reached with a strong, very strong difference between 2 arms, where the Lutathera combination had a 79% reduction in risk of progression compared to the control arm, and with an estimated PFS of 40 months compared to 8.4 months with high dose octreotide therapy. On top of that, there was quality of life measures showing improvements in overall, as well as some specific points which were gathered in around diarrhea, flushing, as well as abdominal pain. We recently had a full 5-year analysis done on the trial, and still median overall survival continued to show benefit compared to the control arm, even though not statistically significant, but was highly clinically relevant based on how the experts see the data. And on top of that, the most important part was we didn't see any new emerging safety signal after long-term follow-up of 5 years. So since its approval in January 2018 in US, the almost 8,000 plus patients have been treated with this. And on top of that, this platform opens the discussion or further evaluation that can it be tried wherever other tumor where we have a somatostatin receptor expression. And there are a lot of, we already have a pediatric study going on, even though this tumor, neuroendocrine tumors are rare in pediatric, but we have a paraganglioma, pheocytochromosoma, and some other tumor types where we are exploring this further. And also studies like NETTER-2, where we are even studying it in earlier line, in first line, in tumors, high proliferating tumor, where in the grade 2 and grade 3 tumors. So with that, we see a lot of interest in last 3 years from physicians in terms of exploring in number of different tumor types expressing somatostatin receptors. So back to you, Brian. Thank you.
Thank you. That's a very nice overview and a lot of really interesting points that have that you bring up there. In fact, I'd like to say that all of you have brought up a number of things that are that get my mind thinking about where are we headed? What are the next? What are the things that we're on the brink of? Much like the poll that I asked at the very beginning. If I could ask, Rodrigo, one of the things that a lot of us experience who are in oncology now and have developed these very targeted, very innovative agents. For example, a pathway inhibitor, as you're describing. When we talk to patients or when we're talking to the public and trying to explain how this work is important, how the foundation of science moving forward is there. What have you found? What lessons have there been in the development of QINLOCK that you think you could leverage to try to increase public understanding or awareness of the innovation process behind medicine?
So thank you, Brian, for your question. I think this is an important aspect. One of the things that we have been doing at Decipher from the beginning was to engage with advocacy groups, in this case with GIST patients. So we have been working with the Lateral group, for example. We have working with the groups in Europe in order to understand their needs and how they see our drug when it was actually in clinical trials. And the reception was very positive because, as I mentioned, there were no treatments approved after the standard of care. So being able to provide something else for those patients is really good because you're going to continue allowing these patients to continue living for several months, hopefully years while on treatment.
Great, thank you. Thank you very much. I'd like to kind of put that same question to you, Barinder. Lutathera is in the radionuclide therapy. Again, as we look to the public and public understanding and awareness of radiation, there's that immediate radiation bad mindset. But we know that radiation can be good, and the more targeted and the more focused the radiation, the better it is. So again, could you share on that same point how to get to a better public understanding, or what were some of the challenges in trying to get from your early proof of concept into human trials and ultimately being approved and ultimately being adopted, in that increasing the understanding and awareness of what you're trying to do?
No, thank you. It's a very, very important question, even from the radioligand space. Exactly what you mentioned, that radiation is a taboo if it's not explained well. So there is a major initiative going on through same advocacy groups and directly through other means where we really differentiate how radioligand therapy is a targeted therapy. It's a precision target where it's not the whole body radiation the way we treat other tumors. On top of that, I think the most important part is also the whole idea around how strong or different the data is compared to when you treat the tumors from the safety perspective with the whole body radiation. So those type of discussions are very important in this. But I think our initial experience is very good from patient perspective, because I think our challenge is less of that, more of the logistic part, because you can only treat these patients in some type of centers which are ready for a radioligand therapy treatment. So we have seen patients on their own used to fly to get the treatment. Initially, when it was not approved in the US, patients were flying to Europe to get the treatment. I mean, just to give a little story, the way it initially started, because as most of you might already know, this product was originally part of AAA, or Advanced Accelerator Application, which was acquired by Novartis. And just to give a little history, this started as an academic exercise at Erasmus Medical Center in Netherlands, where they actually were compounding the treatment and giving it as compassionate use, and which formed the basis for going to FDA to do a phase 3 study once AAA acquired the rights to develop it commercially. So our major challenge, as we are expanding in the space with new indications, you might have seen the PSMA data in the prostate, late stage prostate recently presented at ASCO, is going to be how can we, because this is not off the shelf drug. So this is more of a treatment which is manufactured when the order is placed, and the logistics, the life side, the half-life of lutetium, the decay pattern and all that. So you need to give certain treatment. So a lot of effort is going, and I can say that patients understand all these logistic challenges and why this radiation therapy is not same at other, but we have to continue to give this education as we move along.
Thank you. Thank you very much. That's really insightful. You know, one of the things that you just mentioned about the kind of the origin of the therapy and starting in academia and then in an accelerator before then kind of moving into the hands of Novartis there, it brings up an interesting thought. You know, the Galien is focused on innovation, and a lot of the innovation that takes place occurs in biotech companies or smaller companies. My primary focus, of course, is the biotech world and the development of agents through there. But it is a misconception with the public that all drugs come from big companies. So Jason, I'd like to call on you for a moment. You know, we have Novartis represented here, we have Bayer represented here, we have these global names, but we also have Barinder from a company that not as many people are familiar with, or you from Blueprint. Talk to us a little bit about the challenges of being not just the challenges, but I would say the challenges and the advantages of being in a sphere where you're working in a smaller company, and how you get from that early proof of concept stage to where you are now.
Sure, yeah. I'd say one of the major advantages and part of why I joined Blueprint was honestly the focus that is required at a small company. So you tend to have a very clear plan, you know, the therapeutic areas you want to go into, the types of targets you want to work on, and with that focus just comes kind of the commitment from, you know, the lab all the way up through, you know, development, commercial, and the board, that these are the programs that are important to us, and then everyone can work with a sense of urgency without wondering about when their program is going to be cut due to a portfolio decision, et cetera. So I think that's, you know, and as a result of that, you know, I think a lot of the people that come to a company like Blueprint, they're all interested in that. And so everyone comes here to roll their sleeves up, you know, reach into other areas. There's, you know, no sense of, you know, it's not my job. You know, you can reach anywhere you want to move these therapies forward. It's also part of why, of course, we focused on precision oncology. It's a very attractive area for a small company, because if you're selecting the right patients and you're bringing forward high quality molecules, you're going to see these patient responses even as early as dose escalation cohorts. So you very quickly know that you have a promising drug on your hands, even in those early studies. And then again, it gives you, more conviction to move that compound rapidly through the clinic to help these patients. Does that help?
Great.
Yep.
Yes, very much. Thank you. I appreciate that. So Ian, I want to turn to you because in your discussion, you brought up something that I think is really important in the world of innovation, of precision medicine. And that's that the drugs we're developing for some patients, they are a single agent that works alone. But we know that cancer is very complex and there are resistance mechanisms and everything doesn't work for everybody. So you mentioned that Bayer is taking Nubeqa in the direction of combination therapies and trying it in different settings within the disease. I'd like you to talk with us a little bit about that, about kind of some of the benefits of trying to combine that way, some of the challenges as you're trying to reach across the aisle, so to speak, and work with a company that's developed a different technology or is going in another way. And now you're sharing and looking at things together.
Yeah, I think that's an excellent question, and certainly there are a lot of opportunities and a lot of challenges when you're looking at combination therapies, and particularly in areas within oncology where there are already a number of agents approved. As we've seen in recent years, there are tumor types where for decades there was a standard treatment approach, and all of a sudden that treatment approach is one of many, or may not even be an appropriate treatment approach anymore. And so the opportunity there is, you can potentially take advantage of additive or synergistic effects with other agents to potentially have a greater impact for the patient long term when you do have proven therapies that are available for the patients. The challenges, are a few. One of them is when there are multiple therapies already available for a patient, the challenges of doing clinical trials for those patients are potentially significant, right? Because a lot of physicians, a lot of patients may be satisfied with the therapies they have. So then how do you move forward? And the way you move forward, there are a couple, right? I mean, typically the standard paradigm, as we know in oncology drug development, is you work initially in patients in clinical trials for patients who have limited therapeutic options or no therapeutic options, and you gradually move forward towards earlier disease. But prostate cancer, even at the point when this drug began to enter its late stage trials, there are already therapies approved for more of the later stage patient populations, which is one of the reasons that the initial trial for this drug wasn't in the most advanced patients. It was in an earlier patient population, in patients who had prostate cancer that had spread, but not prostate cancer that had spread to the point that you could detect it with standard imaging. So that's what we call the non-metastatic castration-resistant prostate cancer patient population. So these patients had already received standard first-line androgen deprivation therapy, and then went on to be randomized to Nubeqa or others. But certainly, as you then look at other patient populations where there are more established treatment options, the best approach is really to combine, right? And to look for the best partner, and to look for situations where you're not going to have overlapping toxicities, where the tolerability profile hopefully is not much worse than it would be with one agent on its own. And you're looking for that synergistic or additive benefit for those patients. So the great opportunity is that you have the potential to have one course of therapy that provides the patients more benefit than they would receive with therapy A or therapy B when you combine the two of them. So that's the great opportunity, but certainly, challenges with tolerability there potentially, and challenges in terms of, differentiation, if you will, in terms of, why this therapy versus another, which, obviously is important as you think about clinical trials and, recruiting patients and having patients and physicians comfortable with the new treatment approach.
Great, thank you. Thank you. It's very, very interesting and a lot of good points there. So I'd like to talk to you guys as a panel for a moment, and I have essentially the same question, I suppose, for all of you. You're representing again the general field of precision medicine and the future of our discipline, which is these more tailored treatments. So I'm going to ask you to get your crystal ball or whatever and try to look to the future, and share with me where your thoughts are. As scientists and as clinicians, where do you see the next wave of innovations trending in your particular product areas, and really what are these new areas of science in the fight against cancer that excites you the most? So I'm going to go alphabetically, and I'm going to start with Barinder.
So thank you, Brian. Very important question. So I think from the radioligand therapy, I think this is a very new platform. As I mentioned, this is a first FDA-approved treatment, the Lutathera space, but it's expanding. And a couple of areas where we really need to think of, can we do basket trials with multiple tumors expressing same type of receptors and get bucket bucket kind of approval. So that's one way to look at. The other thing is how can we make, even though it's infusion-based treatment, if we talk about lutetium-based treatment, still you might see some toxicities or the safety events. How can we make it even more safe to remove those barriers? So one of the things, and then other thing is patients are still failing even after these treatments. How can we find solutions around different type of radio, for example, alpha emitters? So lutetium is a beta emitter. So how can we go to alpha emitters and retreat patients with those type of isotopes? Finally, right now, in a bulky tumor, you can use beta emitters, but there is also thought process that can radioligand therapy good for low expressors? I mean, we will think that you need to have a tumor highly expressing the receptor to kill the tumor cells. But if we think in a flip manner, if the radiation has a reach beyond that, so beyond that one cell, having with a beta emitter, can we have a low expressor tumor also get benefit from radioligand therapy? So this is a totally different way of thinking, because we always combine precision medicine with the highly expressing a target, and we totally block it. So the other thing is, can we combine radioligand therapy with the already approved novel target agents, but you need a very low dose because you are not actually using that target treatment, approved treatment for killing the cell. You're actually using only as a carrier because the killing is done by isotope. So you can remove all those toxicities with things which are fully approved already, and you're giving it a very low dose. So those are the things which we are thinking from the radioligand space in future, in addition to how can we make it all safe and also around that.
Thank you. Thank you. So Ian, I guess the same question to you. Where do you see the next wave? What might the Galien committee be looking at in 5 years?
Yeah, well, I think what I would say first is I see a new wave of collaboration extending that's going to produce those potential opportunities. And certainly in my career, I've seen the level of collaboration extend dramatically. Certainly, we see collaborations, partnerships between small companies and large companies, such as those represented here today. But even more importantly, with academia, with patients, with patient advocacy groups, caregiver groups, as well as funding organizations such as venture capital, which is an area which is exploding today, certainly in the Boston area where I live, and elsewhere in the US. And so I see more of that happening. And what that's going to do, I think, is going to create a variety of different therapeutic advances of many different types. I mean, we've seen a variety of different approaches all progress dramatically in recent years. So I think we're going to see precision oncology approaches, targeted therapies, be they small molecules, large molecules, the antibody drug conjugates, targeted radionuclide therapy, as Barinder was saying, cell and gene therapy. We've seen a lot of work there, including but not limited to new CAR T options for patients. And then I guess also I'm expecting the next wave of immuno-oncology therapies to add on to the great advances that have happened there. And I think, you had asked me, the question earlier about combination therapies and such things. I think how to combine all of these is going to be a great challenge, but it's going to be a great opportunity at the same time. How you figure out how to combine them, how to sequence therapies is a really challenging area right now. And so it'll remain challenging, but I think it's a great challenge to have, because what it means is there are more therapies available for the patients who need them.
Great, thank you. So Jason, what about you? What's exciting you? Where do you think we're headed?
Sure. Yeah. So three areas come to mind. Number one is just overcoming the traditional limitations of precision therapy, and by that I mean sort of durability and treatment resistance. And I think we're making some strides there. Being a little bit more thoughtful upfront around on-target resistance mechanisms. So what could the cancer cell do to mutate the protein of interest to stop the effectiveness of your drug? But then there's bypass mechanisms, and having a better understanding of those in the patient populations, and being a little bit more forward thinking around what sort of combination approaches could be useful as other pathways are activated. The second one is what I would call tumor agnostic, which I think Barinder alluded to with the basket trial comment. And again, for the audience, so that's just, regardless of the tumor's location in the body, as long as a disease driver is present, can we, not think about it as lung cancer or thyroid cancer or bladder cancer, but a cancer that is driven by a specific mutation or protein that's activated. And so I think we've been making progress there, but I think continuing to evolve with the industries and the FDA's thinking around how those trials are run and how the approvals work there. And then finally, just expanding the scope of targetable cancer drivers in precision medicine. So as we've heard today, and as we've seen for the past decade, precision medicine is just a great way to have a dramatic impact on patients' lives without the toxicities associated with chemotherapy and classical radiation. But we're going after certain classes of targets, and there are plenty of known drivers where we could select patients, but we just don't have effective ways to target so-called undruggable proteins. And again, the industry is doubling down there. There's quite a few strides, as everyone knows, the past couple of years on targeting certain mutations of KRAS. And again, I think as an industry, we're just going to continue to go after those undruggable targets, since we know the promise of precision therapy if we can drug them.
Great. Sure. All right, Rodrigo, I'm going to have you back clean up here. What do you think is coming up? What might we be seeing in the next 5 years, 10 years? What's exciting to you?
So, honestly, I believe that QINLOCK will continue making noise, and I think that you guys will continue seeing data from us. So the treatment landscape for GIST is actually changing a lot. There were a couple of compounds that were approved last year. There are several compounds in development. And in particular, for QINLOCK, we have an ongoing phase 3 clinical trial moving to the second line. Actually, it's a head-to-head versus sunitinib. We're anxiously waiting the results. I know that the GIST community is also eagerly awaiting those results, because that could potentially really change the landscape for the treatment of GIST. There are also, as many of my colleagues have mentioned, combination therapies are important. We believe that combining a KIT inhibitor with another compound that will be targeting the pathway is really important. We have good preclinical data when combining with a MEK inhibitor. So actually, we're going to be starting a phase 1/2 trial in combination with Kinlock and a MEK inhibitor that's going to have to start before the end of the year. So really excited that we can really move that maybe to the front line versus imatinib. We don't know yet, but hopefully that's going to continue bringing hope for patients with GIST.
Wonderful. Thank you. Thank you. Well, we're almost at the end of our time together. So if you all recall at the beginning of the webinar, I asked the audience to give their opinion on what they felt would be the most promising medical advance likely to happen in the next 5 to 10 years. So thank you. We've got our results up there now. So it looks like there was kind of a split introduction of comprehensive diagnostics and therapeutic vaccines, and then the widespread use of patient-friendly digital and biosensor platform technologies. Thank you. So again, very interesting results we have there. I'd like to thank Barinder and Ian and Jason and Rodrigo, all of you guys. I'd like to thank you for your time today, for sharing your knowledge, your experience, and your insights on where we're headed in the solid tumor field, as we're trying to build these new therapeutic options to not just to fight cancer, but to address the prevalence and to address these really vulnerable populations. I'd like to remind all of you that are watching or listening that this webcast will be viewable on the Galien Foundation's homepage. And just a reminder to join us for Thursday's forum, the Pre-Galien Awards event, and presentation of the 2021 Roy Vagelos Pro Bono Humanum Award, recognizing the companies that led to this year's unprecedented early arrival of vaccines and therapeutics for COVID-19. Again, thank you all, and I will adjourn the webinar.
Thank you.
Thank you.
Thank you so much.